So today I'll be talking about fluorescence resonance energy transfer, which is called FRET in short. My name is Zainab Ahmed, and I'll be talking about background information, principle, application of technique. I'll be going over a short case study and a question. So background and introduction. So this technique was discovered by Theodore Forster and was published in his paper in 1946. It is dependent. The energy transfer non-radiatively from an excited molecular fluorophore donor to another fluorophore, which is called an acceptor. Intermolecular long-range dipole-dipole coupling. Reduction in the donor's fluorescence intensity and excited state lifetime, and an increase in the acceptor's emission intensity. The absorption or excitation spectrum of the acceptor must overlap the fluorescence emission spectrum of the donor. The donor and acceptor transition dipole orientations must be approximately parallel. The most important factor is the proximity of the donor/acceptor pair. Now we're going to be talking about the application of technique. The donor fluorophore typically emits at shorter wavelengths that overlap with the absorption spectrum of the acceptor molecule, which may be a fluorophore or non-fluorescent molecule. So FRET is used as a tool for identifying the distance relationship and supramolecular organization of cell surface molecules. Explosive increase of FRET applications in biomedical research is seen today. One way of multiplexing using organic dyes is involved in sequential FRET between donor and acceptor fluorophores, where excitation of one or more donors results in a cascade of energy transfer. And then it's also used as quantum drop FRET. Quantum drops are inorganic semiconductor nanocrystals, and they exhibit unique photophysical properties such as broad absorption bands and narrow emission spectra that can be tuned by varying the size of the nanocrystal. And then the quantum drop FRET technique takes advantage of their donor properties, coupling them with multiple acceptors. Such systems have been applied to DNA hybridization, reaction, cellular imaging, and monitoring of enzymatic activity. And then on the right, you can see the picture of a FRET. What happens is like, so as you can see, the blue fluorescent protein, it transfers its energy to the green fluorescent protein. And yeah, that's basically how a FRET works. So for this, we're gonna be looking at our article called Single Molecule FRET Imaging of GPCR Dimers in Living Cells. And so for this article, they use SM-FRET, which is also referred to as single pair FRET. It's a robust tool for reporting distances less than 10 nanometers between fluorescent probes. The approach has the molecular scale resolution needed to directly measure GPCR complexation. Its use in mammalian cells has rarely been reported due to multiple experimental and data analysis challenges. The most notable studies use SM-FRET to study conformations of proteins labeled in vitro and microinjected into cells. It's reported advances for SM-FRET imaging that enable investigations of receptor dimers in living cells, including agonist-induced structural dynamics between promoters. So for the data analysis, which is this figure, so we can see that all the way to the left, it's a set of control points used to derive a nonlinear transform function t. The spatial position of the donor signal was then calculated by mapping the acceptor position at each point onto the donor channel. In the middle, we can see the representation of SM-FRET in sequences of SFMGLU2 diffusing at the cell surface, showing the acceptor during FRET and its corresponding donor in the left and right channels, respectively. Trajectories for the acceptor and donor are shown as red and green lines in the image sequence. On the very right, you can see fluorescent intensity time traces were generated, and FRET was calculated for each SM-FRET trajectory. The time traces correspond to the images sequence and SM-FRET trajectory shown. And then, so from this, you can theorize that with only donor excitation, it's observed that acceptor's intensity that collides with that of the donor with that of the donor indicative of the detection of receptor complexes by SM-FRET. Donors without colocalized acceptors are consistent with the stochastic labeling approach employed, in which a population of dimers labeled with 2 donors or only 1 donor and no acceptor is possible. Using SM-FRET, it shows that MGLU-R2 forms stable dimers at all receptor densities explored. Finding indicate that the dimerization prosperity of some GPCRs may greatly depend on local concentration and thus differ across tissues and macrodomains.
So my question is, what do you think are some drawbacks of using FRET in biomedical science?